Today we'll be discussing about carbohydrate metabolism. As we know, carbohydrates are the major source of energy for all the living cells. Monosaccharide glucose is a central molecule for carbohydrate metabolism. All the major metabolic pathways are connected with glucose. Glucose is utilized at the core source of energy. If we see here, glucose can be converted into many pathways. If we go to the oxidation pathway of glucose, we can get glycolysis, that is M pathway, HMP shunt, uronic acid pathway, also TCA cycle. It can be stored as glycogen. That process is known as glycogenesis. It can be converted into fats, lipogenesis, can be converted into amino acids and other carbohydrates also. If we see the major pathway, what is the major pathway of glucose metabolism? Carbohydrate metabolism. One is glycolysis, another one is TCA cycle. Glycolysis. What is glycolysis? So, if we break the name, glyco plus lysis. So, lysis means breakdown. Lysis, that means breakdown, and glyco, lysis of glucose. By what? By oxidation. Oxidation of glucose or glycogen to pyruvate and lactate is called glycolysis. When pyruvate in case of when oxygen is there, and lactate when there is no O2, in absence of oxygen. This was described by Emden-Meyerhof pathway. Glycolysis is also known as. If we see the salient features of glycolysis, what is the significance of glycolysis pathway? It is the only pathway that is taking place in all tissues of our body. Glycolysis is the only source of energy in erythrocytes. In strenuous exercise, when we are doing heavy exercises, muscle tissue lacks oxygen. That means we are going our muscles are going through an anaerobic condition. Anaerobic glycolysis forms the major source of energy for our muscles. The glycolytic pathway may be considered as the preliminary step before complete oxidation, because we can see later that after glycolysis we are getting pyruvate. That pyruvate enters in the TCA cycle after converting into the acetyl CoA. So after the completion of TCA cycle, that is the complete oxidation. So if there is no glycolysis, we cannot get acetyl CoA that further go in the TCA cycle. So glycolysis is the preliminary step before the complete oxidation. Glycolytic pathway provides carbon skeleton for synthesis of non-essential amino acid as well as glycerol part of the fat. Most of the reactions of glycolytic pathway are reversible, which are also used in the gluconeogenesis that we see in the subsequent videos. What is gluconeogenesis? By means of time, you know that gluconeogenesis means new glucose formation. Neogenesis means newly formation. So formation of glucose from non-carbohydrate source is known as gluconeogenesis. Now we are directly moving into the steps of glycolysis. So these are the steps of glycolysis. This you have to remember. So what is our source? That is glucose. Glucose is converted into glucose 6-phosphate. with the help of ATP and magnesium, and enzyme is hexokinase. That this step is irreversible. Glucose 6 phosphate to fructose 6 phosphate. Here the enzyme is phosphohexoisomerase. Isomerase because glucose and fructose are isomers, so they are getting just converted into another form of glucose, that is fructose. Glucose 6 phosphate to fructose 6 phosphate, and the enzyme is phosphohexoisomerase. Then come to the third step. Third step is fructose 6 phosphate to fructose 1,6 bisphosphate. Another phosphate is getting attached with fructose 6 phosphate, fructose 1,6 bisphosphate, with the help of ATP and the enzyme phosphofructokinase. This step is also irreversible. Then fructose 1,6 bisphosphate to glyceraldehyde 3 phosphate and dihydroxyacetone phosphate with the help of enzyme aldolase. This glyceraldehyde 3 phosphate, when converting into 1,3 bisphosphoglycerate, one NAD plus molecule is helping it, and NAD plus is converting to NADH plus H plus, and the enzyme that helps in the fifth stage, that is glyceraldehyde 3 phosphate dehydrogenase. Then 1,3 bisphosphoglycerate to 3 phosphoglycerate. One phosphate is coming out, and that is in the form of ATP, energy. Here we are getting energy, and the enzyme is 1,3 bisphosphoglycerate kinase. 3 phosphoglycerate to 2 phosphoglycerate. Just the position of the phosphate is changing here, and there the enzyme is phosphoglyceromutase. Next step is 2 phosphoglycerate to phosphoenol pyruvate, and the enzyme is enolase with the help of ion magnesium Mg2 plus. Phosphoenol pyruvate is converting into pyruvate with the help of pyruvate kinase, and that is another irreversible step. That is the end product of our glycolysis pathway, pyruvate. So here in glyceraldehyde 3 phosphate, so we are getting glyceraldehyde 3 phosphate and dihydroxyacetone phosphate. From that one molecule of glyceraldehyde 3 phosphate we are getting pyruvate and dihydroxyacetone phosphate can further converted into glyceraldehyde 3 phosphate. It's a reversible pathway, that glyceraldehyde dihydroxyacetone phosphate can again convert into glyceraldehyde 3 phosphate. That glyceraldehyde 3 phosphate again converting into pyruvate. So if we take one molecule of glucose, from one molecule of glucose we are getting 2 pyruvate. So what we have to remember in the steps of glycolysis, full step of glycolysis with the enzymes, and where the ATP is gained, where the ATP is reduced, where NAD plus NADH plus H plus is produced, everything we have to remember. Important point is the irreversible steps of glycolysis. That is first step, where the enzyme is hexokinase. Third stage, that is where the enzyme is phosphofructokinase, and the ninth stage, where the enzyme is pyruvate kinase. What is the rate limiting step of glycolysis? Where the enzyme phosphofructokinase is activating, this is the rate limiting step of glycolysis. Now come to the next stage, pyruvate as a junction point. Why we are calling pyruvate as junction point? Because from pyruvate it can be directed to various pathways. What are the pathways? That pyruvate, as we can see, is converted into lactate. When it is converting into lactate with the help of lactate dehydrogenase, when oxygen is not there enough, in anaerobic condition, pyruvate is converting into lactate with the help of lactate dehydrogenase. Besides that, it can be decarboxylated to acetyl CoA, which enters the TCA cycle, or that acetyl CoA can be used as fatty acid synthesis. Pyruvate may be carboxylated to oxaloacetate, which helps in the gluconeogenesis that we can see in the subsequent videos. But for now, you have to remember that pyruvate is a major junction point from where various pathway can be started. After converting acetyl-CoA, it is going to the TCA cycle. That acetyl-CoA helps in the fatty acid synthesis also. And pyruvate, when it is converted into oxaloacetate, from this oxaloacetate, we are going to the steps of gluconeogenesis. What are the regulations of glycolysis? If you see the regulations of glycolysis, that is hexokinase. Hexokinase enzyme, phosphofructokinase enzyme, and there are some allosteric inhibitors or allosteric activators. So hexokinase is inhibited by glucose 6-phosphate. Hexokinase, go to the step, hexokinase and glucose 6-phosphate. You can easily appreciate that when glucose 6-phosphate is more, it will inhibit the hexokinase to convert the glucose into glucose 6-phosphate. So for hexokinase, glucose 6-phosphate is an inhibitor. Next comes the phosphofructokinase. As I have said, phosphofructokinase is the rate limiting, you can say it is a rate limiting enzyme. That step is rate limiting step of glycolysis. Allosteric inhibitors are ATP and citrate, and allosteric activator is AMP. Now come to the enzymatic control. When we are concerned with glucose, there are two major enzymes that comes in mind. One is insulin and another one is glucagon. So when we are in fed state, we have just eaten, we are eating. In that stage, insulin comes forward, and insulin is the enzyme that is active now in the fed state. That favors the glycolysis because you are eating, glucose sources are there in heavy amount, so insulin helps in the glycolysis and glucagon inhibit glycolysis. Now come to the energy yield in the glycolysis. So we are coming to the steps, that is if you see the steps of glycolysis, you can easily appreciate that in the first step ATP is used, in the third step ATP is used. So when we are calculating energy, we can say that in the step of first step hexokinase, 1 ATP we have reduced, and in the third step also minus 1 ATP. Then come to the fifth stage. Here 1 NAD plus molecule is converted into NADH plus H plus. From 1 NADH plus H plus molecule we are getting 2.5 ATPs. So for 1 NADH we are getting 2.5, and what I have told that dihydroxyacetone phosphate is again converting into glyceraldehyde 3-phosphate, and that is again converting into 1,3-bisphosphoglycerate. For that another NADH is produced, so there total 2 NADH plus H plus molecule is produced, so total 2 NADH, 2 into 2.5 we are getting 5. And then in the 6th stage we are getting 2 ATPs, and in the 9th stage we are getting 2 ATPs. So there are 4. So 4 plus 5 is equal to 9, and here we are getting minus 2. So 9 minus 2, 7. After the end of glycolysis till the pyruvate, we are getting 7 ATPs in the aerobic condition when oxygen is available. And if we see when oxygen is deficient in the anaerobic condition, in anaerobic condition there is no production of NADH plus H plus. In anaerobic condition, NADH plus H plus is not produced. So for that, in the first step is similar, we are getting that one ATP molecule is reduced. In the third step also one more ATP molecule is reduced. After that 2 and 2, 4 ATP molecule is also produced. So 4, and from there we are getting minus 2. So 4 minus 2. In anaerobic condition we are getting total 2 molecules of ATP after the glycolysis. Now there is an important cycle, that is Cody cycle or lactic acid cycle. So it is a process in which glucose is converted to lactate in the muscles, and in the liver this lactate is reconverted to glucose. What is happening here? When pyruvate is converted into lactate in the anaerobic condition, so when we are going through exercises, our muscles are not getting enough oxygen, so then pyruvate is converted into lactate. That lactate is easily permeable through our cell membrane, so it is coming in the blood. Lactate is coming in the blood. This lactate is going in the liver. In the liver, with the process of gluconeogenesis, lactate is converted into pyruvate, and from pyruvate it is converting into glucose by the process gluconeogenesis. That glucose is again coming to the blood. So this cycle is known as Cody cycle. What is happening here? That pyruvate going through the anaerobic condition and it is producing lactate. That lactate is coming in the liver. In the liver, lactate is converting into pyruvate. That pyruvate is converting into glucose by the help of gluconeogenesis, and that glucose is supplying energy. This is a very important chart because here we can see what are the major pathways can happen after the glycolysis from this pyruvate, from this glucose. So glucose, phosphoenol pyruvate and this pyruvate, that is the process of glycolysis. From pyruvate you can get lactate in the anaerobic condition. From pyruvate this alanine can be produced. This is amino acid. From pyruvate acetyl CoA is produced with the help of enzyme pyruvate dehydrogenase. From that acetyl CoA it is going to the TCA cycle, or it can be used as a fatty acid synthesis. Now this pyruvate has different fates. So what are the fates? Fates of pyruvate: lactate, alanine, that acetyl CoA that is going in the TCA cycle. This is pyruvate carboxylase enzyme. That pyruvate carboxylase enzyme is converting pyruvate into oxaloacetate. That oxaloacetate is, with the help of phosphoenol pyruvate carboxykinase enzyme, is again converting into the phosphoenol pyruvate. From this it is going to prepare glucose, that is the gluconeogenesis, and that oxaloacetate can again help in the production of this aspartate or malate, major amino acid. So what is converting pyruvate to acetyl CoA? That is pyruvate dehydrogenase complex. Why it is complex? Because it is made up of multi-enzyme complex with 5 coenzymes and 3 apoenzymes. So it is known as pyruvate dehydrogenase complex. Under aerobic condition, pyruvate is converted to acetyl CoA, which enters the TCA cycle to be oxidized to carbon dioxide, and ATP is generated. So that is all for today. What we have learned: the major pathway of carbohydrate metabolism, that is glycolysis. What is happening here? Glucose is converting into pyruvate. In that process, that can we are getting ATPs, so energies. So glycolysis pathway, you have to remember all the enzymes where the ATP is produced. That ATP yield, how it is going in the oxygen in the aerobic condition, in anaerobic condition. What is the regulation of glycolysis? So this you have to remember. And there is a cycle called Cori cycle or lactic acid cycle that is happening in the anaerobic condition. So that is all for today. Thanks for watching.